So I'm Carl Barrett. I lead the Translational Sciences group at AstraZeneca. And the title of this session is Effectively Integrating Biomarkers into Study Designs. So I used to be the head of the biomarkers and imaging group at Novartis, and I grew to hate that word because everybody uses it in different contexts. And so I prohibited anybody from using the word biomarkers without putting a qualifier in front of it. So you're talking about a safety biomarker, predictive biomarker, a pharmacodynamic biomarker, or a surrogate endpoint. So this session is actually pharmacodynamic biomarkers, so we're very clear. We had a good discussion this morning about sort of why drugs fail. And usually when people talk about, and it certainly was the case this morning, drugs fail because of either efficacy or safety. In some ways, neither of those are, both of them are right and neither of them are informative, right? Because obviously cancer drugs are always toxic, and so you have to actually get an effective dose that's not toxic, but actually it hits the target to the degree with which you want to hit the target. And so that's really what pharmacodynamic markers are about. How hard can we hit the target? First, we need to know how hard do we need to hit the target. Is it a matter of, we made a lot of mistakes in the past because we thought 50% inhibition was correct. So the Plexicon drug, which is now approved to the rapamib for BRAF melanomas, would have failed if they hadn't been bought by Roche and they reformulated it and actually got twice the exposure and got 90% inhibition versus 50% inhibition as it was originally. So PK/PD, hitting the target, and again, hitting it not only hard enough, and we oftentimes underhit it, but also long enough. And so again, sometimes sometimes it's better to hit it hard for a short period of time. Sometimes it's hard to hit it. You want to hit it for a long time. Again, a lot of mistakes were made in always developing drugs for daily dosing. We now know that intermittent dosing, you can get higher doses, maintain your safety margin, and get greater efficacy. So PK/PD is a huge area of unmet need in the pharma company, and I think an opportunity, as we discuss this morning, for dog development. So I'm really looking forward to the presentation. So the first is by Tim Phan, who will talk about opportunities for preclinical evaluation of novel therapies. Tim.